Dear honorable viewers and all the students of MHC in entomology, Assalamu alaikum wa rahmatullah. Welcome to the discussion on cuticular sense organs of insects. If you've already subscribed in my channel, thank you very much. If not, please subscribe now to get the notification of next video. For this, click on the subscribe button below the video. Then click on the bell icon and then finally select all the option all. Very important questions on this discussion is: describe various cuticular sense organs of insects and mention their function. The sense organs or receptors are those structures whereby the energy of a stimulus arising outside or within the insect is transformed into a nervous impulse, which, after transmission to one of the central ganglia, usually results in the change in the behavior of the insect. Sense organs are associated spatially with the integument, and each organ or sensillum typically comprises: number A cuticular structure through which the stimulus is translated into other mechanical or chemical changes, and number B, one or more sensory neurons responding to the modified stimulus resulting from nervous activity. All the cuticular sense organs under the following 6 main headings are discussed. Number 1, Mechanoreceptors. Number 2, Auditory Organs. Number 3, Chemoreceptors. Number 4, Temperature Receptors. Number 5, Humidity Receptors. And number 6, Visual Organs. It may be noted that auditory organs are actually only specialized types of mechanoreceptors, while humidity perception may ultimately prove reducible to one of the other mechanisms. Number 1, Mechanoreceptors. The members of this group have the common property that they are excited by processes involving the mechanical deformation of some parts of the receptors. 4 main structural types of mechanoreceptor may be distinguished. Number A, articulated sensory hairs. Each sensory hair comprises The usual trichogen and termogen cells, which secrete respectively the hair and its socket together with a bipolar neuron, which is produced distally into specialized structures containing arrays of microtubules and in close contact with the base of the hair. Such hairs are widely distributed over the insect body, especially on the antennae, tarsi, and cerci, and are tactile organs being stimulated to produce a nervous impulse on movement of the hair in its socket. Number B. Trichobutria, singular Trichobutrium, are elongate city hairs present in arachnids, various orders of insects, and myriapods that function in the detection of airborne vibrations and currents and electrical charges. In 1883, Frederick Dull observed that they were deflected by the sound waves from a violin and labeled them hearing hairs. Number C. Camponiform sensilla. This consists essentially of a dome-shaped region of a relatively thin cuticle, often oval in surface view and set a little above or below the general level of the integument, with a denser rim of cuticle surrounding it. These sensilla occur on various parts of the body, including the cerci, wings, and legs. These respond to the external pressure. Number D. Chordotonal organ, Scoloparia. These are usually compound structures composed of a number of specialized sensilla, scolophores or scolopedia, which each contain a relatively conspicuous sensory body, the scolops or scalopel. Each chordotonal organ consists typically of a spindle-shaped bundle of scolopophores attached at each end to the integument, but less frequently the distal attachment is absent and the organ ends freely in the body cavity. These respond to the stimulation of pressure, movement, and touch. Number 2, auditory organs. They help in the detection of sound vibrations transmitted by the tympanic membrane and cause vibration of the hairs of the tactile sensory. Four types of sound receptors may be recognized. Number A. Tympanal organs. These are paired structures, always composed of a thin cuticular membrane, the tympanum, associated with tracheal air sacs and chordotonal sensilla. They occur in the Orthoptera, the Cycadidae, and some Lepidoptera. In the Acridoidea, there is a tympanal organ on each side of the first abdominal tergum, with an externally visible tympanum surrounded by a cuticular ring. Internally, a group numerous scolopopores form a swelling known as Moller's organ applied to the inner surface of each tympanum and connected by the auditory nerve to the metathoracic ganglion. Number B. Auditory hairs. Munich, 1925-1963 has shown in many lepidopteran larvae that responses to sound are mediated by hair sensilla, which react to low frequencies. Number C. Johnston's organ, Roth 1948, has shown that in male of Aedes aegypti, sound wave can set in motion the hairs on the antennal flagellum, and this in turn causes movements of the whole flagellum, which stimulate the sensilla of Johnston's organ. Number D. Pilifer of Chirocampine hawkmouths. A unique auditory organ sensitive to ultrasonic frequencies has been found in the head of several species of Sphingidae. In these insects, sound perception depends on contact between the median wall of the second segment of the labial pulp and the distal lobe of the pilifer, which contains the sensory transductor. Number 3. Chemoreceptors. Chemoreception is developed to a remarkable degree in insects, particularly the taste or gustatory sense of smell or olfactory sense and senses responding to common chemical stimuli. Number A. Gustatory senses. Taste. These are defined by responses to a relatively high concentrations of non-volatile stimulatory substances which usually come into contact with the receptors in aqueous solution. For this reason, the gustatory sensilla are sometimes known as contact chemoreceptors, and they mediate important responses in feeding, the rejection of unpalatable substances. The receptors include trichoid basiconic sensilla. Trichoid sensilla is hair-like and basiconic is peg-like sensilla. Number B. Olfactory sense, smell. The sense of smell in insects is stimulated by low concentrations of the vapor phase of a great variety of substances which are relatively volatile at ordinary temperatures. The olfactory receptors are trichoid, hair-like, basiconic, peg-like, coeloconic, sunk in a pit, and placoid with a mere plate sensilla, and are found in antenna and labial bulbs. Number C. Common body chemical sense. In insects, helping them to avoid irritant substances like acids, secretions in the environment, the receptors have been suggested in some cases to be peg sensillae. Number 4. Temperature receptors. Heat may be transferred to or from an insect by radiation, convection, or conduction. These methods are important as stimuli to behavioral changes. Specialized areas on the body of acridity have been identified as thermoreceptors. Number 5. Humidity receptors. Insects respond to changes in humidity to water vapor, and humidity receptors have been described on the antennae of Tribolium, pediculus, etc. In the former, they are the thin-walled branched beaks, and in the latter, they form sensory hair organs. Number 6. Visual organs. Responses to light are mediated by number A. Dermal receptor, number B. Dorsal ocelli, number C. Lateral ocelli, and number D. Compound eyes. Number A. Dermal light sense. The body surface responding to light, as in the larval tenebrio when all light receptors are occluded. Number B. Dorsal ocelli. There are 3 dorsal ocelli on the frons and vertex of the head, though the median one may be missing. Each is composed of the cornea, the corneagen layer, the retina, pigment cells, and central nervous connections. These respond to light on the absence of compound eye. Number C. Lateral ocelli. They are located on the side of the head. In some groups, there is a single ocellus present on either side, while in others there may be 6, 7 or more ocelli. In the Tenthridinidae, the single lateral ocellus is not unlike the typical dorsal ocellus in structure. The cuticle forms a lens-like cornea secreted by a thick underlying layer of corneagen cells. Beneath the latter is a retina composed of many retinulae, is made up of 4 cells whose apposed rhabdomeres forms a rhabdome. A broadly similar type of structure is found in tipula. Number D. Compound eye. The principal feature distinguishing compound eyes from ocelli is that in the former the cornea is divided into a number of separate facets, whereas There is only a single facet to each ocellus. Compound eyes are aggregations of separate visual elements known as ommatidia, each corresponding with a single facet of the cornea. After Ames General Textbook of Entomology, 10th edition, page 123 to 160. Thank you very much for watching and keep watching for the next video.